Hello, everyone. We're going to start very soon. Welcome to today's seminar series called Cutting Edge Advances Towards Personalized Mental Health, hosted by our Center for Precision Mental Health and Wellness. I'm Dr. Xue Zhang, and I'm a co-director of this seminar series, along with Dr. Leonardo Tosi. I will kick off with a couple of housekeeping items. This talk will be 40 to 45 minutes with additional 10 to 15 minutes for Q&A. It is now being recorded and will be posted on our website next week. If you're interested in this recording or previous seminar talks, you can find our website in the chat box or scan the QR code on the left side of the screen. From there, you can also sign up to receive announcements about our future seminars and events. If you're interested in joining our center as a research member, please see the link in the chat as well, or scan the QR code on the right side of the screen. For Q&A, for the in-person attendees, please raise your hand if you have a question. We will ask Dr. Shinozaki to kindly repeat the question for the people on Zoom. For virtual attendees on Zoom, please put your questions in the Q&A box, and we will attend to them at the very end of the presentation. I encourage you to put your questions as early as possible because they will be addressed in the temporal order. Now I hand over to the director of our center, Dr. Lian Williams, to give the introduction of our keynote speaker.
Hello, everybody. I am Leanne Williams, Director of the Stanford Center for Precision Mental Health and Wellness. Today, it's my great pleasure to introduce our special keynote speaker, Dr. Gen Shinozaki. Dr. Gen Shinozaki is an Associate Professor of Psychiatry here at Stanford University. He completed his psychiatry residency and psychosomatic medicine fellowship at the Mayo Clinic in Rochester, Minnesota. His research interests, as you'll hear, include a focus on novel EEG methods and algorithms and device development for detecting delirium, predicting outcomes, and developing novel therapeutics. Before joining the Stanford faculty, he worked closely with Dr. Jimmy Potash, the current chair of Hopkins Psychiatry Department, and who was his mentor for his K23 award. Dr. Shinozaki currently holds an R01 for an NIMH and is leading a clinical study for novel EEG devices to screen for delirium. And his methods have been validated in over 1,000 patients. He has received, not surprisingly, prestigious awards, including the 2019 Glynn Fisher Clinical Research Award and the Visiting Professorship Award from the Academy of Consultation Liaison Psychiatry. So welcome, Dr. Gen Shinozaki. We are delighted to hear from you, and you'll be presenting on game-changing approach for delirium and your development of bispectral EEG devices, integrating with epigenetics biomarker mouse model for detection, prediction, and novel therapeutics. Welcome, and I'll hand over to you.
Thank you very much, Dr. Williams, and everyone in the team. And thank you for this great opportunity. And I'm very excited to share my passion on my delirium research. Should I share my screen? I think. Okay. Right. Yeah, so because of time limitation, I decided to focus on two parts of three topics, my EEG device development and also epigenetics biomarker. If time allows, maybe I can get into some of my preclinical work using animal model, but we'll see how it goes. Okay. So the first part is my passion about EEG device development, which I call bispectral EEG, BSEEG. It is developed to detect delirium, but not only that, we found that that can be very powerful to predict patient outcomes. Okay. So I have already published related papers, a couple of papers already. So if you're interested, please refer to them later. When I give this talk, I like to start with question. Why do we measure blood pressure in the hospital? Or how about glucose? And because we have an in-person audience, I like to seek a volunteer. Why do we measure blood pressure in the hospital? Or how about glucose? Any volunteer?
In the hospital, they're proxies for worsening disease or worsening state.
Yeah, so brave volunteer kindly share her thought. Like we are using that number to identify high-risk patients so that we can help them promptly, right? That's true. Like if patient has a high blood pressure over 200, like hypertensive emergency, we should respond and treat them. If I have a patient with low glucose, they are at risk for life, so we need to treat them. So that's correct. So please come back to this question later, okay? Okay. And next, I want to borrow your imagination, right? So let's say your grandma or grandpa or mother or father, or depending on your age, let's say your loved one is admitted to the hospital. You'd hope that she or he would come home safely, okay? But it's not really easy because of the delirium. So there is a large literature says that delirium is common and dangerous. But unfortunately, usually it's underdiagnosed, and because of that, it's undertreated. So there has been a lot of effort to try to screen delirium, but those are unfortunately not practical at this point. And because those are not practical, it's not widely used in busy clinical practice. So I said that delirium is common. Some estimates say that over 12 million elderly people admitted to the hospital annually in the United States, but among them, 20 to 50% in general medicine floor, or up to 53% after surgery, or in case of ICU, close to 90% develop delirium. So at least 2-3 million cases of delirium happen every year, which is a huge number. And I said that delirium is dangerous and also expensive. Why? Because delirium is confusional state, so they can lead to higher incidence of complication. Like they can fall, they can injure themselves. Not only that, because of confusion, they can punch nursing staff and injure nursing staff, and they can aspirate the infection. So because of those complications, they tend to stay longer in hospital. Some estimates say that more than 1 week stay longer. Unfortunately, notoriously in the United States, one-day hospital stay costs over $2,000. So 1 week longer stay can cost easily like $15,000 more. The worst thing about delirium is that is associated with high mortality. One study shows that more than 40% people die after 1 year. This is almost as bad as the mortality after heart attack, right? But in case of heart attack, we do a lot of things. But for delirium, we haven't done that much. And because of above mentioned complications, they tend to be discharged not to home. Instead, they go to nursing home or rehabilitation places. So the overall cost for one case of delirium can be over $60,000. And in the previous slide, I said that 2-3 million cases happening every year. So total financial loss can be $150 billion per year in US alone. The same number is true for Europe and Japan and China, probably. So Dr. Sharon Inoue used to be at Yale, now the famous researcher in Harvard, has published a lot of paper, delirium in older persons, New England Journal of Medicine, delirium in elderly adults, in Nature Review Urology, delirium in elderly people, Lancet. I wish I learned how to do this, so many similar papers in the major journals. But joke aside, and Dr. Inoue is very nice to me, so I shouldn't tease around. But the point is, she has been putting lots of effort to around the society. We should pay attention for delirium. We should do more. But unfortunately, nothing has changed that much. So this is one of the examples I mentioned. Famous paper over 2 decades ago. Patient with delirium is dotted line die 40% after 1 year compared to non-delirium people. And this fact has been replicated by many, many other independent research, including my own data, that I will show you later. So then there has been a lot of effort to screen and detect delirium. But current screening tools can be like confusion assessment method developed by Dr. Inoue herself in 1990, which is over three decades ago. Then later on, ICU version called CAM-ICU, which has been widely used in hospital to some extent, developed by Dr. Wes Eli, intensivist from Vanderbilt. And other thing could be delirium rating scale, more non-specific screening can be MMSE or MOCA, etc., etc. Some information says that there are like 40, 50 different kind of delirium screening to exist. So even for our specialist, we as a psychiatrist, we should be specialists, but we cannot catch up with those. So the problem is those tools need to be used by human beings. Otherwise, it doesn't count, right? So this example, like Minicog, CAM, and also this CAM-ICU, this was made like a nice colorful flowchart with the hope that it's easy to be used by busy hospital staff. But in reality, they are too busy to implement that, you know? Before coming to Stanford, I worked at the University of Iowa. Then when I was working on clinical study, I had a chance to talk with the head of nursing managers. So I think like nurse, like a leader, something like that. Then I asked her why we don't implement this delirium screening in the hospital. And her reply was that she said, we just failed. And I asked her back, what do you mean by you just failed? And she said that she tried to implement screening hospital-wide, but the busy hospital staff never could catch up. They couldn't implement that. So they just implement only in ICU, which was okay. So that's the reality in nationwide. Then I thought about EEG. Okay, why? Because traditional EEG can be detected, can detect delirium. Okay, but it's not unfortunately practical for every patient, even with high-risk patients, because the machine is too big, as you know. And then technician need to place multiple electrodes all over the head through sometimes sticky glue through the hair. And then finally, neurology specialists need to interpret that. So although I'm a big fan of ordering EEG for my patient in a hospital, but if I order EEG every day for every patient, neurologists wouldn't like me, right? So that's the challenge. But when I ordered EEG for the suspected delirium cases, the finding would be reports say that diffuse throwing, non-specific. But what do you mean the diffuse throwing? It means that throw wave like delta or theta happening across diffusely all 20 channels. Okay. Then I saw that if all channels are showing the same signals, we don't need 20 channels. Maybe the Q channel could be enough. Then actually this concept has been, you know, like proven by my colleague from Netherlands, like almost 8 years ago, using 20 channel every combination. Then he showed that only 2 channels can be enough. So this is an example of normal EEG, and this is an example of diffuse throw wave. Okay, and can you see this cursor? Yeah, this is normal and this is diffuse, right? And can you tell the difference? Okay, so this is again volunteer time. Okay, how about top one? Is this slow or normal wave?
Slow.
Yeah, slow, right? Then how about the middle one? Yeah, then how about the last one?
Very slow.
Yeah, yeah. So slow, normal, slow. So now you are expert in EEG reading, right? So the two channels is enough. But actually, this fact has been known since 1940s. Engel and Romano published famous paper. Top one is marked delirium. Below one is normal awareness. Just 2 channels can tell the truth. Okay, but this knowledge has not been implemented. Or actually, I realized in other areas of medicine, they were using it, like anesthesiology, monitoring for depth of anesthesia. That machine is called the BIS monitor or entropy, also a brand name. They are using it for measuring the depth of anesthesia so that patient will not wake up in the middle of surgery, which would be a bad thing, right? So I heard that has become a kind of standard of care for anesthesiologists for the last 2 decades or so. And even our own area of psychiatry, when I was a resident ECT rotation, I saw the ECT machine and we put 2-channel EEG to monitor the duration of seizure activity. Then I thought that we should bring this machine to my hospital round to capture the delirium. But you know, the ECT machine is at the end, it's a little bit too big to carry around. But that was the concept. So this is the example of BIS monitor. So, you know, putting an electrode on the forehead, and then with 30 seconds or so, you get the number like 98. So then anesthesiologist tried to keep this number below 50 or so, right? So obviously I was awake at this point, right? But you know, so I thought this is pretty good. But I think this machine itself is this size, it's a little big to carry around. Okay, so I thought maybe I should do by myself the delirium EEG study to test if a simplified EEG device can detect delirium among elderly high-risk patients. And also at the same time, I was a little bit ambitious. I want to test if a simplified EEG device can detect delirium before clinical identification. And I'll get to this point later. So the concept is, do you want to do this 20-channel EEG or like do simple device like this, right? Okay. So the study design participant, we did a study, prospective observational study, no intervention, then simple EEG device monitoring twice a day, and we invented digital signal processing algorithm. Then at the same time, we collected clinical data, DRS, DOS, and CAM-ICU twice a day. Then I recruited these patients over 4 years from 16 to 19 up until before COVID hit. Then initially we started with 2 cohorts of high risk for delirium, orthopedic surgery patients, and also older adult general medicine patients. Then over 4 years, we recruited over 1,300 subjects, and the positive for delirium was roughly 30%. Then 70% was control non-delirious patient. Then 2/3 came from general medicine, 20% came from orthopedics, and later on we added patients from ER and ICU. Then female and male was roughly half and half. Then average age was 68. So initially this was our first device. I call it the palm size device. This was kind of finicky device, but eventually gave us good data. So put electrode on the forehead like this, where I had more hair before. And then this is a cartoon version. Like for the sake of data gathering, we started with 2 side channels, right and left. I was not brave enough to put my own picture in my paper, so I just used cartoon. Maybe I could have. But. So for the sake of data gathering, we started with 10 minutes recording right and left. So the top one is like a raw EEG data. Then we did a free spectrum density analysis, right? So by doing that, you can capture how much power into each frequency. So based on that, we tried a bunch of different algorithms, then came up with the algorithm, what we now call the BSEEG score. So a BSEEG score, higher score meaning more slow wave, then lower number meaning less slow wave, right? So when we compare the case of delirium with green dotted line and a control case, a control patient with black triangle, we were able to differentiate the two. Not only that, when day 2 and day 3, when delirious patient become really delirious, score picked up, meaning more slow wave. Then when patient recover, score went down to the normal range. So we are very happy about this start. But as you see, this is one case on control. So we need to do more, which we did. So initial 45 samples, we divided delirium case and control. Then with reasonable cutoff, and we were able to achieve AUC 0.70. As you know, this is not the greatest AUC, but it's a good start. Then sensitivity over 80%, specificity 80%. Let me close, I can see this. Let's see. I should do this, okay. Then, but I need to validate this. So what I did, let's see. The additional 24 inpatient cohort, AUC was 0.81, sensitivity to specificity over 83%. When I brought this to ER, over 100 patients, AUC 0.84, specificity 91%, specificity 77%. Then when I collaborated with a Japanese electronics company called Fujitsu, they are able to help me to implement a technology called TDA, topological data analysis, and that can definitely enhance our AUC over 8 to 0.84, over 137, 145 subjects. Okay. Then now next, we were hoping to get much smaller device, which I call the thumb size EEG device. And even with this, when we try to do DRS, which is delirium rating scale, so which can quantify the severity of it, we can see the association with BSEEG score in a dose-dependent manner. Okay. And I said that I was a little bit ambitious. I'll come back to this point. So the blue and the green was like DRS and DOS, so the clinical measurement. Then orange one is EEG score. So there's no picked up. This is negative case. Then this is a positive case. The DRS and DOS picked up in day 6. So this is the timing when clinician can identify patient with delirium. But orange one, the EEG score picked up 2 days earlier. If we can prove this, potentially we can intervene, we can identify high-risk patient even before clinical identification, then potentially we can intervene. So we have great opportunity to help them. So far, I hope that I was able to show that EEG can be helpful to detect delirium. But why do you care about detecting delirium? Because as I said, because that leads to poor outcome. Then my next question was, can EEG capture outcome? That was my next question. I tested that and it did. The length of stay and BSEEG score were significantly correlated, adjusted for age, gender, and CCI. CCI is a Charlson Comorbidity Index, which is a good indicator for severity of comorbidity predicting outcome mortality. Then discharge outcome was also associated. But the real outcome we want to care about is mortality, as I said, right? So this is a refresher, like 40% of delirium die faster, right? Before going to that, I want to make sure that my clinical team's assessment for delirium are good enough to replicate those famous findings, and it did. Our clinical team definition of non-delirium, delirium, people with delirium die faster. So that was correct. So our clinical definition was good, doing well. But the other question was, can EEG do this? And it did. EEG score low, meaning less slow wave. BSEEG score high, meaning more slow wave. They die faster. And you see this formula. I put day one EEG, meaning that this is the first time EEG we got collected when they came to the hospital. Just like 3 minutes, 5 minutes recording, then you can predict who's going to die faster in 1 year, which is scary almost. Yeah. So this is a comparison: clinical category, no delirium, delirium. EEG category, less slow wave, more slow wave. You can pretty much replicate clinical judgment. So I'm joking around. When I do too well with this technology, I may lose my job. Like a joke aside, the nice thing about this BSEEG score is different from dichotomous diagnosis yes or no for delirium. This is a more continuous variable. So you can divide into more stratified way, low, middle, and high. Then you can stratify the risk, mortality risk, which is plus. Not only that, this is interesting, my favorite slide. So when we combine clinical diagnosis and EEG categorization, purple one, they were judged to be clinically delirious, and EEG was positive, meaning slow wave. So they die fastest, no surprise. The blue one was clinically judged to be delirious, but EEG was saying that you're okay. They didn't die faster. It's almost as good as orange one, who are clinically judged to be okay, and EEG was fine. Not only that, this green one, they were clinically judged to be okay, but EEG was saying that something bad is happening in your brain. They die faster. This group of people at this point with clinical practice, we cannot identify, because those are the people who can answer memory question, orientation question correctly. Without EEG, we cannot find those high-risk people. This is important and also scary. We are missing those high-risk people in the hospital every day. And I believe that probably our brain is capable of covering up something bad is happening, but superficially in conversation, they can answer some superficial good answer correct question, but EEG, electrophysiological change, we can identify. This data is pretty consistent with my dear colleague, Dr. Kimchi from Harvard now in Northwestern, who showed inpatient mortality with EEG, delirium diagnosis, and he clearly showed that EEG is more important than clinical diagnosis, which is kind of sad as a clinician, but okay. I was puzzled, can EEG be that good? So I asked my student in Iowa to go back to the EEG suite and collect the data led by neurology specialists, then capture people who had a EEG report of diffuse slowing and normal. Then this is the outcome. Again, clearly people who had EEG diffuse slowing die faster compared to normal group. So technically, to some extent at least, our one-channel EEG can replicate what neurology specialists predicted by a formal reading EEG. Not only that, this is fun part, not fun, but dementia and delirium is hard to distinguish, especially diagnosing delirium among dementia patients whose baseline cognitive function is compromised is very challenging. We psychiatrists pretend as if we could do it, but not in reality, it's hard. So when we combine the BSEEG delirium, dementia diagnosis and BSEEG, no dementia group, still we can differentiate the mortality risk, but especially among a dementia patient, if their BSEEG is okay, they're okay. But if their BSEEG is bad, they die really, really fast. So, potentially if we can implement this technology to nursing home patient, we can identify high-risk patient early on. So, another example of application of BSEEG, sepsis diagnosis is very tricky. Lot of objective number like a vital sign, like saturation, blood pressure, respiratory rate, then altered mental status, which is very fuzzy and subjective. I didn't like that. So among sepsis patients, when I applied the BSEEG numbers, we were able to clearly differentiate the stratification of mortality risk based on BSEEG score alone. And this has been replicated with my small sample size EEG device as well. And by normalizing everything together, now over 1,000 patients, we were able to replicate. Based on BSEEG number score, we can predict mortality. And especially among delirium patients, we categorize everybody as delirious, but their mortality can be totally different between BSEEG score. Okay. This is a little bit more specialist question, but among delirium, there's a subtype called, motor subtype called hypoactive delirium and hyperactive delirium. Hyperactive delirium is kind of common delirium patient, like agitated, putting IV, punching, nothing stuff. It's easy to be identified. Hypoactive delirium is withdrawn, not responding, can be regarded as easy patient, quiet patient. But inside the brain, something bad is happening. Among them, still, BSEEG score can differentiate their mortality risk. So regardless of motor subtype, we can predict who's going to be at high risk. Okay. So coming back to your grammar in the hospital, I said that we measure blood pressure and glucose to identify high-risk patients. Based on these data, I believe that we should check BSEEG as well. We should monitor BSEEG to identify high-risk patients and try to intervene. Okay. So brief summary for this talk. Part, delirium is dangerous condition and early detection is vital for better outcome, and current methods are not practical. So simplified EEG can detect delirium early and easy to use for busy hospital settings. And this approach would benefit patient, physician, hospital, and health economy. And future direction, I envision this BSEEG score to be used as a next vital sign, used every day, every patient in the hospital, clinics, and nursing home. Okay. And at some side, newer device being tested. And the real goal is to bring this technology to the patient. Okay. So that part is done with EEG. And should I take a question now, or should we go to epigenetics part? Maybe I should go. Yeah. Okay. All right. Okay. So totally changing a shift. I hope I won't confuse you, but because before starting working in device developing study, my K grant was epigenetics of PTSD. Okay, so like NIMH is funding me. Don't tell people, but NIMH is funding my 80% time. But I really enjoy device development for EEG. So the PTSD epigenetics and device development EEG, and I felt kind of torn apart in between. But I enjoy both. But at the same time, I thought maybe I should think about combining between the two. And then when one day typed epigenetics and delirium, there was only one hit. And even one hit was talking about like dementia context, not focusing on delirium at all. And it was not really talking about what I was thinking. Because I came up with some idea, then that's what I'm going to talk about. And it became very fruitful. Okay. So I published a couple of papers about this already, so please refer to them. Okay. So among epigenetics, as you may know, there's various different mechanisms, the DNA maturation, histone microRNA, but in my simplest mind, I focus on DNA maturation. Okay, it's easier to understand. We believe that the maturation happening on the gene can cover up the gene promoter region, especially suppress gene expression. So that's the key concept. Okay. All right. So then, as I said, delirium detection current approach is limited. EEG is helpful, but current one is not practical. That's why I'm doing BSEEG. But the bottom line, we need a good biomarker. Right? Then thinking about delirium risk factor, that's aging and also infection surgery. But in reality, in reality, I mean, not pathophysiology is not really well understood. Okay, then I got the inspiration from animal model. There's a lot of animal studies showing that cognitive disturbance happen especially among aged animal after like a lipopolysaccharide, LPS injection or surgery. Okay, then they show that neuroinflammation is a key, Nowadays, we talk of neuroinflammation everywhere, like depression, schizophrenia. But I believe that delirium is extreme of neuroinflammation. Anyway, so what did neuroinflammation mean? I mean, more cytokine elevation, right? Like pro-inflammatory cytokine, interleukin beta, interleukin 16, alpha. Then it's coming from microglia activation. Then cytokine release from microglia are enhanced among aged animals exposed to exogenous insult. So that seems to be the key in mice model. So then I learned the term about microglia priming, which I didn't know before. So young mice, microglia is resting state, then going to primed state, then going to activated, then release cytokine to interfere with neurotransmission. So it takes some time. But in case of aged one, microglia is already primed. That's why it's easily to be activated, then release cytokine a lot, then mess up the neurotransmission, which makes sense to me. So there's a lot of animal studies showing that with stimulation, younger age, there's a lot of cytokine elevation, microglia activation. So it sounds like a well-proven concept. Then in a human, it's known that heightened inflammation is happening in human. Of course, mainly in blood, but interleukin 6, CRP, and other cytokines, more and more. Unfortunately, data has been kind of inconsistent, but kind of overall, the inflammation, elevated inflammation in delirium patient is well accepted. Then I thought about this, and I thought maybe in a human delirium, maybe microglia is similarly primed and activated. I call the microglia hypothesis, meaning apply the microglia in aged animals. It could be the same in priming in microglia in elderly patient. Then that's why maybe cytokine is heightened in elderly patient with delirium, like already primed, that's why it easily get activated and release more cytokine. Then I thought about epigenetics. Why? Because if aging changes expression rate or is like a readiness for expression, something in controlling expression mechanism should be changed. And I was wondering what is that? Then I thought that could be epigenetics, especially DNA methylation. Why? Because as I said, DNA methylation control expression of the genes. And so if I call it epigenetics hypothesis of delirium, if microglia priming happens with aging, and if enhanced cytokine release from microglia is true in human, expression control of those cytokine genes may be regulated with epigenetics change, which is likely happening along with aging. Because aging and DNA mutation is a well-known relation, has a very long, well-proven relationship. Among epigenetics mechanism, DNA mutation is known to change dynamically along with aging and throughout lifespan. And the transcription regulation with DNA mutation is well known. So I thought in microglia, cytokine-related genes may be regulated through DNA mutation, then especially with aging, that's changing. So that's why enhanced cytokine release in response to exogenous stimuli happen, then leading to delirium. So that was my hypothesis, but I need to prove it. So that's the data. Well, before going to data, so the cartoon concept of aging can be enhancing epigenetic change, then that can be leading to microglia priming, then exposure to exogenous insult, enhanced cytokine release happens, then cognitive disturbances. It's a well kind of accepted concept other than epigenetics part in animal model. So in case of delirium, high cytokine level is known after surgery or infection, there may be microglia priming happening through epigenetics change from aging. So this equal part is already proven, right? We know the similarity, but we are missing link with this yellow link. So that's what I want to test. So the data. So the approach I took was using Illumina genome-wide Illumina EPIC array method. The old version is 450K, now newer version is called EPIC version. I think they stopped using EPIC version 1, then they started setting version 2. So EPIC version, we can check 850,000 CpG sites across genome. The concept is that along with CD, methylation tend to happen in cytosine followed by G guanine called the CpG site, as you may know. So when methylation is happening on a cytosine, after bisulfite conversion, kind of chemical conversion, methylated cytosine remain as cytosine, but unmethylated cytosine is converted to uracil, leading to T. So this array compares the difference of C and T with the ratio. So that's the way we measure the level of methylation level. Okay. So initially what I did was I asked my friend from Emory, who has a Grady-Tron project, which has a large number of cohort from poor community with a lot of DNA methylation data. So simply I asked them, please check cytokine gene, candidate cytokine gene, and their age correlation. I asked them like interleukin beta, interleukin 6, interleukin 8, and TNF alpha. Then please sort them based on the strengths of the strengths of like correlation along with age. Then to my surprise, only TNF showed up to the top. And these are p-value 10 minus 12 minus 11 minus 11 minus 10, all negative correlation with age. And this one only showed, I showed GTP, Grady-Tron project, but actually this is from a PGC, a Psychiatric Genetics Consortium PTSD EVAS group. So they have different cohorts like Detroit trauma cohort and Detroit Center cohort. And the exactly same trend have not trend, same signal from three cohort. And I invited other two groups to publish together, but they said that they don't want to do it. So I just use the GTP, which is the largest, which is okay. So the point is, TNF alpha DNA methylation goes down along with aging significantly. And p-value 10 minus 12 minus 11, you don't see this number with psychiatric genetics, as you know. So I was surprised. So this is a cartoon version or figure version. So along your age, every single CPG goes down. And the good thing about GTP was they did have expression data. So they more highlight. So along with aging, DNA methylation decreases. Then on the other hand, expression increases along with aging. So that's very consistent with what I hypothesized. Along with aging, epigenetics change, especially DNA methylation change. That's why TNF alpha gene is less covered by methylation. That's why that is more prone to be expressed. And the expression data confirms that finding. I was very happy about seeing this, but you may say that this is blood sample, right? You're talking about glia. So next, I want to check using my neurosurgery samples because I have been collaborating with neurosurgeon collecting brain tissue. And then not only brain, these are brain dissected from neurosurgeon for treatment refractory epilepsy patient. Then because sample size is limited, N is only 6, but not only whole brain, but we do fact sorting in glia and neuron part. Then to my surprise, only glia parts showed all negative correlation, but not in neuron, right? So the same TNF alpha gene, even with N equals 6 sample size, the top one is even like nominal significant level. Everything is clearly negative correlation. So that was confirming or supporting what I was hypothesizing in glia. Then you may say that, hey, this is not delirium, right? So next I need to test with delirium samples. So because I was recruiting a lot of patients for EEG study. So after 1 year, I should have realized sooner, but I started collecting biosample as well. So I started collecting blood tissues. Then we did epic analysis, epigenetic study analysis, age and gender matched, starting with over 40 patients, control and case and control. Then I started to focus on TNF alpha because that was showing good signals. Then sure enough, patient with delirium showed a stronger negative correlation, but not in non-delirium control. So we were able to see the difference. This is a figure version. Like red one, delirium patient remained significant decrease along with aging, but not in green non-delirium control. Okay. But as I said, I use a genome-wide approach. So we had more than TNF alpha gene. So next we did enrichment analysis. As you may know, like enrichment analysis among top hit, what genes work together to make sense biologically. So that's what we did. The top hit with a gene ontology pathway was like this. Let me enlarge. The top hit was immune response and FDR significant level. Cell activation, immune response, cell activation. I think the data is too good to be true because just collected 40 subjects, like comparing case and control. But exactly the blood biomarker based on epigenetics is supporting our hypothesis that inflammation immune response is different. And KEGG pathway, similarly, cholinergic synapse, circadian entrainment. The cholinergic function is vital for cognition and delirium, of course. And also delirium feature characteristic is a loss of sense of day and night. So circadian entrainment makes total sense. Okay. The remaining challenge is the samples came from general medicine cohort. The etiology of delirium can be infection, surgery, or other thing. So the tricky side is the etiology can be different. That can confound our signals. So that's general in delirium research. In that case, we tend to use post-operative delirium because the surgical insert trigger for delirium is relatively similar. So that's what we did. And then focusing on post-op delirium sample, because as I said, I was collaborating with neurosurgeons, so I was collecting blood and saliva, other things before and after surgery. So we identified a case of post-operative delirium after neurosurgery, and then compare those similar data. Then that's what we found from independent neurosurgery sample was, again, immune response, T-cell activation, toll-like receptor signaling pathway, cellular signaling. Totally independent inpatient cohort identified signal was replicated in totally independent neurosurgery cohort. Again, for your reminder, let me show that this is an inpatient data from inpatient cohort, immune response. This is, again, some data from neurosurgery cohort. Again, immune response, cell activation, all the same. Limited sample size, but we were able to show that. Okay. Not only that, when we found EOD, which is epigenetics of delirium, our first inpatient cohort, we found genome-wide significant signals, several of them. Then when we compared those signals in neurosurgery cohort, that became more interesting. Pre-neurosurgery is before neurosurgery, post-neurosurgery, after neurosurgery, POD, negative POD, positive meaning no delirium, yes delirium. Before surgery, no difference. All of them, but after surgery, there's a difference, and the directional difference is all the same with EOD inpatient cohort. To make it simpler to view this in a figure, this is like this. Still, it's hard to read, so let me highlight. So before surgery, no difference, but after surgery, we see the difference. Post non-POD, non-postoperative delirium, post-postoperative delirium. The difference is same with inpatient no delirium and inpatient delirium. Again, this has been same every CPG site, just like every CPG site, it was same. Okay. And actually, this paper was recently reviewed but rejected. I don't know why. The reviewer said that was publishing, but somehow editorial decision said that it's not worth publishing. I sent a rebuttal comment, but I will see. So conclusion, we show delimitation differences both at gene and network levels between delirium case and control, and evidence information related to genes and more. And this finding indicates that delimitation status in blood may become a useful epigenetic biomarker for delirium. But not only that, as I said, as I mentioned in hypothesis, I believe that the epigenetics process may play a key role in pathophysiological mechanism of delirium. This is my fantasy, perioperative protocol in 2030. Pre-surgical sample taken from patient, blood delimitation measured for risk prediction, and based on BSEEG score assessed. Then after surgery, we collect blood again, post-op BSEEG for risk monitoring, and then we can follow the chain, then we can identify high-risk patient, then intervene early. Or maybe I should say one more thing. It's interesting was, I'm so obsessed. So the post-neurosurgery, maybe this cartoon is good. So this is a post-neurosurgery sample, we see the difference. But both pre and post sample were collected in OR, an operating room. Before surgeon cut the first incision, we collect pre, and after they close the scar, we collect the post. So technically, patient hasn't even waken up. The patient hasn't left the OR. So technically, they haven't even exhibited any clinical symptom. But epigenetics marker in the blood has already shown that who are at risk for developing delirium. Current technology takes like 2-3 days to get this data, but we can assemble a new technology to get this score in like OR. We can pay a lot of good attention. So, all right. And a happy grandma back home. That's our goal. That's why we are doing this. Right? Okay. And I appreciate funding, NMH and SF, Sumitomo Farm, some industry collaborator, and So, I had a lot of students in my lab in Iowa. I built a team, and then a lot of them went to the hospital to recruit patients, like 1,000 patients. Crazy, right? I mean, before COVID, we reached the peak that we were recruiting maybe 25 patients per week. So if COVID didn't stop that, probably we could have added 1,000 more in 1 year. But anyway. And then many of them went on to medical school, grad school, residency, and faculty positions. I'm proud of them. And I moved with one postdoc 2 years ago, and I'm now rebuilding a village. So thank you very much.
It's such a wonderful talk, and let's see if we have any questions from the in-person attendees.
I have a couple about the BSEEG. Do you think it could distinguish between delirium and some of the forms of catatonia, like an agitated catatonia?
So that's, to begin with, a hard question. How BSEEG can differentiate catatonia and non-catatonia. My traditional view of catatonia is that usually catatonia should have relatively normal EEG. And then for me, diagnosing something has to guide us for treatment or prediction. Then if I make a diagnosis of catatonia, then first treatment would be giving a benzodiazepine for adipant challenge. And that is very effective. But if I know that patient has delirium, I would not give benzodiazepine. So there's some kind of group of my colleagues whom I like, saying that catatonia and delirium can happen together, and I'm still kind of debating if I would buy in or not. So BSEEG, I should be able to differentiate. I think they should be able to differentiate largely with catatonia and non-catatonia. Yeah.
And I have one other. I mean, just as a clinician, I think, with the early detection, I can understand get the anticholinergic off, get the benzo off. But is there any sort of ideas that you have for additive treatment? So something that you give the patient that would be useful to them how to detect them?
Yeah, so actually I'm doing clinical practice now, and then that's a real challenge because technically there's no good treatment. So the question is how to treat after detecting delirium, which is a real challenge because we do not have any proven treatment. And the relatively recent paper, maybe 5-7 years ago, showing that lamellitione, a stronger version of melatonin, melatonin receptor agonist, 3-4 times more potent than regular melatonin, has been shown to be effective to prevent delirium and also potentially treat delirium after onset of first night delirium. There's a mixed follow-up result, but I believe that is effective because that is re-regulating circadian cycle, and that's what I usually go to. And antipsychotics has been widely used to control the agitation for the safety for patient and nursing staff. But there's a lot of literature recently has shown that it's not effective at all, which I believe. So I didn't have a chance to talk about my animal study part because BSEEG in human works so well. So I started applying BSEEG to a mouse model, which showed nice diurnal change and clear response to LPS. So now we can quantify the severity. And also now with that scheme, we can test a lot of new compounds. So that's what we are doing. So what medication can really prevent those worsening EEG? So stay tuned. Yeah.
Thank you.
Mm-hmm.
Questions down there. Thanks so much for coming to talk today. It's super impressive that you're able to advance both the mechanistic side of what's actually going on and then also these immediately translatable clinical outcomes to act on. I guess in terms of linking the two together, is there anything on the slow waves that are specific to the delirium states that you're seeing being the body's response to driving the clearance of the cytokines, TNF alpha and 6 beta that you're seeing pop up in the brain? It is a biomarker, but I noticed that you seem to be intentionally not referring to it as a treatment target, but a marker of, and obviously a very effective marker.
Yeah, so the question, to summarize from my understanding, how specific that slow wave can be, kind of.
Yeah, and I guess, is it potentially happening to clear out?
Yeah. So there's a nice, interesting animal study showing that with hemisphere, one side you give cytokine, the other side you don't give cytokine. Then clearly on the side you got cytokine, got slow wave. So obviously that is cytokine sensitive. So the question is, if we deal with cytokine, could we prevent those slow wave or even delirium, which we do not know. But I'm hoping to test more with this my animal model, and eventually if we can test in a human as well.
We have quite a few questions from Zoom. I can definitely read them. The first one is, can you stratify BSEEG to correlate with the cause of delirium?
I don't think this is that specific. And eventually, yes, I like delirium for study, but eventually we may need to depart from that terminology, because psychiatrists said delirium and the neurologist said encephalopathy, and the ICU staff said altered mental status. And then we discussed in silo. Then the meeting we went to, I went to this week was called American Delirium Society, and more interdisciplinary anesthesiologists, psychiatrists, neurologists, internists get together. Then we always talk about it. So we agree that we need to find a new name. But my personal opinion, maybe we can simply go with acute brain failure, just like acute heart failure. But etiology can come from different group. But my goal is that by using BSEEG, we can find abnormal signals so that we can look into what's causing it. So to your answer, BSEEG is not specific to specific cause, because more like a bracket measurement. That's originally my hope. That is my hope. So I hope that I answered the question.
Thank you. We have another question asking, do you have any data showing a medical intervention based on your BSEEG that changed the outcome with different therapy in ICU?
That's my next goal. I'm hoping to get started with such clinical trial. I'm not yet. I'm starting with animal study for now. So stay tuned. Maybe you can help me.
Great. And the next question is also related. Do you suggest using aging mouse model or inflammation model to study delirious mouse? What's the difference?
Yeah. If it's okay, can I share my mouse study, please? Quick, I guess. Yeah. So we are testing mouse model using let's see, what's the slide show? Yeah, so because this is mouse EEG, maybe stop share maybe. Let's see. Yeah, so brief answer. I mean, mouse. We are testing mouse model, aged mice and young mice, and we can clearly see that this is mouse. So younger mice, so we get a nice diurnal change for young mice, and then 12 hours difference. Average, you get a nice stable change with LPS injection. You get a huge increase, just like a slow wave, right? So the nice thing about it is that you can quantify based on LPS dosing. Then young and age, we compare. Then same dose, aged mice response much worse. Just like older people are more prone to get delirious. So yes, we are testing aged mice.
Thank you. And we have the last question from Zoom. Do you think different anesthesia protocols affect delirium trajectory?
There's a lot of research interest from anesthesiologists. So they are actively looking into that. So far, my understanding that not that much difference expected compared to different from contrary to their original clinical sense. They believe that anesthetic agent, different anesthetic agent can cause different risk of delirium, but my understanding is not so much different. Actually, to be honest, I was surprised to see that a lot of anesthesiologists are interested in delirium research, because I thought anesthesiologists would be so completely done with after surgeries and they don't care about what happened after surgery. But it sounds like they seem to be nice enough to be feeling responsible what happened after surgery, which is good. Yeah. But the one people also say that because in anesthesia, pretty much everything is kind of well established, so they are just like expanding this research field. So, which is good.
So there's another question. I think that's the last we're taking from Zoom, and it's very related to the previous question. It's asking Aspect Medical showed that deep sedation during anesthesia is actually linked to delirium. Did you correlate with the depth of anesthesia and protocols?
Yeah, so that was, I think the group from WashU looked into a lot, like how much depth, or like, yeah. So again, to be honest, there's a lot of mixed report. Data from WashU was negative related to the risk of delirium and depth of anesthesia, but the recent paper from Australia and the Cornell group showed some relevance for that. But I think still there are more to explore, I guess.
Great. Let's see if we have any other questions from our group.
I have a comment. I wanted to echo what Adam was saying before, that I think it's really impressive how you thought about the mechanisms like
Thank you.
neuroscience
Thank you for your kind
and
words.
how that applies clinically, and then also developing the devices to track that.
I'm a bit
And
scattered.
then think about what that means for delivering into a healthcare system and what the impact would be in terms of not only the patient, but the management in the healthcare system. I think that's really rare to be thinking that way, and it's very inspiring.
Thank you very
I
much.
think it's how we think about precision psychiatry in terms of taking measures to access what are changes like the acute brain failure that are hard for a patient to report or for a clinician to observe, and have a way of measuring that. And maybe a speculative question, or asking you to speculate, is given that you found that the BSEEG and the inflammatory marker linking to cognition, at least cognition in a way that links to delirium, could you envision that being applied in other areas? And I asked that from a very different perspective, in that in depression, we see a subset of patients who are younger adults, but they're actually showing quite extensive cognitive impairment. And we're gathering data on whether they're also showing EEG changes. So I'm thinking there may be a wider application of these future vital signs for your 2020.
Yeah, thank you for the kind words and comments. And I totally agree that I was so looking forward to this opportunity because, with Spirit, I think I share my desire to implement more precise mental health care, medical care for patients. In the area of psychiatry, we do not have that much tool set to do it. So this is, I hope, my first step. And I think delirium is the first easy approach. But for example, next approach, I'm definitely thinking about like a dementia. Definitely, I'm pretty sure their baseline is slow wave is higher. But among them, when they get delirious, that could get worse easily. So we know the baseline, we can capture them. Just like a hypertensive patient has a higher blood pressure, but when they're taking medications, okay, but something bad happens, still they go up from there. Depression is challenging for now, because this is very simple EEG, but still, I think those cognitive decline from those symptoms, probably we should be able to see some changes. Maybe you can help me to test this device in your cohort. So please let me know. Yeah.
Sounds good. Thank you.
Thank you very much.
Thanks, Dr. Shinozaki, again for the very insightful comments.